Hi, good morning everyone. Thank you for joining us today. My name is Yvonne from Atlantis Bioscience. In today's webinar, we would like to provide a workflow for stem cells research from culturing of stem cells to analysis. A bit of background of Atlantis Bioscience. We are established in 2016, a private company distributing life science products and lab equipment catered to various states of biomedical and healthcare research in Singapore and Asia Pacific. We provide solutions such as cell culture media, animal serum, cell therapy, microscopy imaging, microfluidics, exosome research, and many more from various partners all over the world. So stem cells have become a very interesting research topic in the recent years. So what are stem cells? They are specialized cells with the ability to self-renew, and they are able to differentiate, giving rise to different types of cells that make up tissues and organs. For example, it can be differentiated into muscle cell, fat cell, bone cell, and so on. So basically, stem cells can be broadly categorized into two groups, the pluripotent stem cells and the adult stem cells. So within the pluripotent stem cells, they can be further classified into the embryonic stem cells and the induced pluripotent stem cells. Embryonic stem cells are isolated from the inner cell mass of the blastocyst, which is a hollow ball of cells that in a human it is formed 3 to 5 days after an egg cell is fertilized by a sperm. So the embryonic stem cells, they have the ability to generate into mature cell types of the three germ lines, such as ectoderm, mesoderm, and endoderm. So for induced pluripotent stem cells, they are basically generated from somatic cells by the overexpression of the four transcription factors, which are also famously known as the Yamanaka factors. So these reprogrammed cells, they are similar to that of the embryonic stem cells, with the ability to self-renew, differentiate, and produce cells of the three germ lines. For adult stem cells, on the other hand, they are multipotent stem cells which are found in various parts of the body, such as in the bone marrow, in the skin, blood vessels, and so on. So these cells are also sometimes called the tissue-specific stem cells, and they serve as a renewable source of specialized cells for tissue development, maintenance, and repair. So here are some examples of the adult stem cells, such as the hematopoietic stem cells, mesenchymal stem cells, neural stem cells, and the endothelial stem cells, and so on. So there are many potential use for human stem cells. They are basically extensively used in the basic research for the study of the normal human development, so that we are able to better understand the abnormal cell division and differentiation, such as in the cancer and birth defects. We are also able to understand the genetic and molecular controls of these processes, so as we are able to develop new strategies for the therapy. Stem cells are also used to test for new drugs. So new medications are tested for their safety and effectiveness. So new drugs can also be tested on how they might affect different organs. Stem cells can also be used for cell-based therapies as it offers the possibility of a renewable source of replacement cells and tissues to treat diseases, which includes diseases such as the macular degeneration, spinal cord injury, diabetes, and so on. So stem cells, they hold the potential to replace damaged cells and organs by supporting the body's intrinsic repair mechanisms to stimulate the human body to generate healthy cells. So now let's take a look at the pluripotency of these stem cells. In fact, for pluripotent stem cell, it actually appears in two different pluripotency states. So in vivo, pluripotent cells are found during a very short window of time in the developing embryos. So let's take a look at this figure. The top is the embryo development for the mouse, the bottom is for the human. So what you can see here, we have the pre-implantation development and the post-implantation development. So basically, the stem cells progress from a naive state in the pre-implantation development to that of the prime state in the post-implantation development. So in vitro manipulations are also necessary to maintain or convert each stage to the other. So now you may be wondering, since both the naive and the prime stem cells, they are pluripotent stem cells, so what are there any differences between these two cells? In fact, there are some key differences that actually differentiate these two. So they have different maintenance signals. For example, in the prime stem cells, right, they are actually dependent of FGF and the MEK signaling pathway, but not that for the naive stem cells. In terms of epigenetically, they are also different. So in the prime stem cells, their X chromosome is inactivated and there is a global upregulation of their DNA methylation, and also they acquire histone trimethylation at their the developmental regulators. So some of the pluripotency markers are also downregulated in that of the prime stem cells, but not that in the naive stem cells. So all in all, the prime stem cells, they are more clonally restricted and have a lower cloning efficiency, and they are more committed to a specific cell fate before the user can direct their differentiation. So now let's take a look at the history of the pluripotent stem cells. Pluripotent stem cells started in 1981, where the mouse embryonic stem cells, they were first derived from the mouse strain 129. This was then followed by the derivation of the human embryonic stem cells in 1998 by Dr. James Thomson, which sparked a revolution in the regenerative medicine. So in 2006, mouse-induced pluripotent stem cells, they were generated. And this is by a technology pioneered by Dr. Yamanaka in Japan. Less than a year later, the same technology was used to derive the human iPSCs, which provides an alternative source of human pluripotent stem cells without the need of using the human embryos. So it is in 2010, naive-like human embryonic stem cells, they were first reported. The naive stem cells these naive stem cells, they were generated by using a transgene-mediated reprogramming of the prime human embryonic stem cells. However, these resulting naive human embryonic stem cells, they require continuous expression of the integrated transgenes for the long-term self-renewal. So in 2012, Dr. Yamanaka eventually received a Nobel Prize for this amazing iPSC technology. So growth medium development for human pluripotent stem cells culture is one of the most critical components of human pluripotent stem cells maintenance. The first generation of the human embryonic stem cells medium, it commonly contained these FBS, fetal bovine serum, and the use of a feeder cells, which is known as the mouse embryonic fibroblast, MEF. However, this poses major obstacles such as the undefined composition in the serum and also the secretion from the feeder cells. Subsequently, FBS was replaced by a knockout serum replacement to generate a serum-free condition. Following that, many defined cell culture systems, they have been developed to culture the pluripotent stem cells. Individual labs and international collaboration have been involved in defining the components in the stem cell culture media. Eventually, this resulted in defined feeder-free and xeno-free conditions. So further progress in the study of the culture media then leads to the formulation of these 8 essential components. However, one important thing to note here is that most of these culture conditions maintain the human pluripotent stem cells in its prime state because of the addition of the FGF in their culture system. So since the discovery of the naive stem cells, researchers have been really interested to establish conditions to maintain the cells in the naive state. So in 2013, naive state culture conditions, they were established. These were by basically using cytokines and inhibitors to convert the primed human embryonic stem cells to the naive state. Although this achieves similar cell culture conditions to that of the naive stem cells, there are significant differences in their gene expression profiles of the naive state markers. There is also a decrease in differentiation potential, and it resulted in unstable karyotypes. So further study then leads to the discovery of a single growth factor known as the NME7AB, which is both sufficient and necessary to maintain the human pluripotent stem cells in its naive state. So these culture conditions provides a more stable karyotypes, and also it increases its differentiation ability. So now let's take a closer look at this culture media. This culture system is developed by Minerva Biotechnologies, a company in the United States who is into the cancer and stem cell therapeutics. So this system is basically serum-free, beta-free, FGF and TGF-beta-free, and it does not require the use of any inhibitors at all. So how it works is that it is utilizing a naturally occurring growth factor, NME7AB, to support the naive state in the human stem cells. So this growth factor is expressed in the earliest days of the embryogenesis, in the inner cell mass of the human blastocyst. This is then later turned off during the development. So what is the mechanism behind this? Basically, it involves this MUC1 star, which is a cleavage protein of the transmembrane protein MUC1. This MUC1 star is actually found on the undifferentiated stem cells, and it stimulates the growth and pluripotency. So basically, the NME7AB, the growth factors, will bind to and dimerizes this MUC1 star, which is found on these undifferentiated stem cells, and then induces and provokes the growth and pluripotency. This culture system involves three components. It involves this single growth factor, NME7AB, and also an antibody adhesion surface that captures the MUC1 star positive cells, and also a synthetic peptide here, which has a sequence of that of the MUC1 star extracellular domain. So basically, this peptide, it acts as a ligand to sequester away the NME7AB. So basically, it breaks the pluripotency signal so that it synchronizes differentiation and ensures that no pluripotent stem cells remain. So this differentiation ligand is basically only added when you want to differentiate your pluripotent stem cells. So one of the advantages of using the alpha stem medium is that it provides an improved iPSC generation. So comparing it to the feeder-based or the M-teaser media where FGF is being used, the alpha stem media is a lot simpler and has a higher cloning efficiency. So using this media, it also allows for large-scale production of the human pluripotent stem cells. So because in the naive state, right, the stem cell expansion is a lot faster, scalable, and it can be completely automated because it eliminates unwanted spontaneous differentiation and is still able to maintain a normal karyotype. So basically within 17 days, you are able to produce make 1 billion cells from starting of a 6-well plate. So most importantly, these naive cells, they are grown in the NME medium, they generate really superior differentiated cells. So here are some examples. The cells can be differentiated into neuronal cells, cardiomyocytes, and the hepatocytes. So this is in comparison to that of the cells grown in the FGF-based media. So as you can see, the cells are a lot more superior. So let's take a look at the iPSC-derived cardiomyocytes here. So this is the video of the pluripotent stem cells cultured in alpha stem media. So as you can see here is that the cells grown in this media have really strong contraction, and the beating is a lot coordinated. And the bottom video is showing the cells grown in FGF media. So the contraction is weaker and the beating is also not that coordinated. So all in all, there are several benefits of using the alpha stem culture media. So it is the first to use a single naturally occurring growth factor. And the generation of this iPSC is feeder-free, and this is a lot simpler and more efficient than that in FGF media. There's no clonal restriction. It provides superior differentiation with improved yield, cell quality, and functionality. The expansion of the cells is a lot faster and scalable. And you can actually passage the cells as single cells, and also it provides stable karyotypes even after more than 70 passages. So now let's move on and look at the another stem cells that has gained tremendous interest, which is the mesenchymal stem cells, or in short, MSC. So MSCs, they are fibroblast-like multipotent adult stem cells. They are derived from multiple tissues, includes like bone marrow, adipose tissue, and umbilical cord. They have the ability to differentiate into different cell types such as the neuronal cells, endothelial cells, fat cells, and so on. So MSCs, they are also promising tools for cell therapy due to several reasons. They are easily accessible as they can be isolated from numerous sources. They are relatively easy to culture in vitro because they proliferate rapidly. They have the ability to differentiate into several different cell types as shown earlier. They also have this special immunological properties and homing ability where they are able to migrate into the site of damage. So basically, generally, they are used for tissue engineering and regenerative medicine. They can be used for vehicles for gene therapy, and they are also used for immunomodulation. So the International Society for Cellular Therapy has proposed minimum criteria to define the MSCs. First, the cells have to be able to exhibit plastic adherence under standard culture conditions. They must also possess specific set of cell surface markers, and they should also be able to differentiate into these three specific cell types: osteoblasts, adipocytes, and chondrocytes. So efficacy, sensitivity, and reproducibility are the key attributes needed for optimal media. Here, I would like to introduce you to the CellCore line of MSC cell culture media products, which is developed by the Excel Therapeutics. Excel Therapeutics is a Korean company formed in 2015 with a team of talented researchers working on the developing media for various cell types. So basically, it begins with the development of a serum-free defined formulation which does not contain any animal or human serum. And recently, they launched a serum-free true chemically defined media which contains materials with non-chemical structures. So the media does not contain any animal or human serum, and the media provides superior proliferation when you compare it to serum containing media. And it supports long-term cultivation and self-renewal of the human MSCs from multiple sources. As being a chemically defined media, it provides consistent and reliable MSC culture. And also, unlike most competitors' media, the CellCore media do not require supplement or any plate coating. So basically, it saves you time and money as you do not need to add supplements or coat your plates or pre-qualify FPS lots. And you are still able to obtain high cell numbers. So you also have the option to choose either the RUO or clinical grade. So let's take a look at the data. So CellCore media, for CellCore media, you are able to get superior proliferation when compared to serum-containing media or that of the competitors' serum-free media. So as shown here, the CellCore media, cells grown in CellCore media are shown in this orange line, and this is comparing to that of the FBS, cells grown in the FBS or other competitors' media. So as you can see here, it has a lower population doubling time, and it is able to accumulate more cells in a shorter time frame. This is the orange one for CellCore. So cells grown in the CellCore media also are able to maintain the standard MSC characteristic based on the trilineage differentiation, and also the expresses the standard MSC surface markers as shown here. So the chemically defined media have also shown to be more stable than that of the competitors' serum-free media. So cells grown in CellCore media shown here in red, they have a lower ratio of cell senescence, They also have a lower, there's also a lower aging, inflammatory, and immune response in the cell core CD media, as shown here for the RNA expression data. So the cells were also tested for serum-related marker, and it's shown in these two figures that they also have a lower expression of the serum marker when compared to the competitor serum-free media. So it is important to keep in mind that although the media performance is really important when culturing MSCs, the need for consistency in culture systems should not be overlooked. So all in all, the cell core chemically defined media, it is able to provide you an ideal culture system to ensure that both the performance and the consistencies are met. So now let's take a look at the matrices, the ECM. Understand that some of you here may still be using culture system that utilizes ECM for the culture of the stem cells and may not be satisfactory due to the cells not growing very well due to low viability and so on. So apart from the culture media, the support of the exogenous adhesion proteins are also very important for the maintenance of these stem cells. Matrix gel is one of the most commonly used matrix. However, it is not well defined as it is isolated from mouse carcinoma cells. So here, I would like to introduce you to a chemically defined protein for the expansion of the human mesenchymal stem cells. This matrix is called the My Matrix MSC, and this is developed by a research team from Dr. Martin von Heusser's lab and the research team from the de novo matrix. So this coating is chemically defined. So it is animal and human component free, and it enhances the reproducibility due to the lot-to-lot consistency. The GMP certifiable coating also facilitates the translation of the research into clinical applications. So this ECM, it is ready to use as a culture where it comes pre-coated in different formats. So therefore it reduces the risk of contamination and saves time as you do not need to coat the dish yourself. So this plates can also be stored at room temperature with safe fridge space, and it is compatible with various reagents and media. So my matrix will be a useful tool for you to use for those who have encountered poor cell attachment or growth when you're culturing your MSC media. So basically, this my matrix MSC is made up of the dextran sulfate and the bone-related ECM peptide 1. So basically, what the research team had done is that they did a screening of the 96 different matrices here, as shown here, and then they identified this combination of bone-related ECM and dextran to be optimal for the expansion of the MSC in the serum-free medium. So this work was recently published in Advanced Biosystems, shown here. So let's take a look at the data here. So my matrix MSC is able to increase the cell doubling by 50% when you compare that to the serum containing or serum-free medium. So my matrix is shown in blue here. So cells grown on the my matrix also show consistent high cell viability. They also maintain their characteristic fibroblast-like morphology and expresses the relevant surface markers for at least 10 consecutive passages and able to differentiate into the 3 specific mesoderm lineages. So another common matrix of interest for stem cells research would be laminin. Laminin 511 is a major component of basement membrane that function as a scaffold for pluripotent stem cells by binding to the integrins on the cell surface. However, laminin is a very, very large protein. It is about 800 kilodalton, which is composed of 3 chains. So usually they form supramolecular aggregates, and it is very difficult to produce as a recombinant protein. So Professor Kiyotoshi from the Osaka University, they solved this problem by producing a recombinant E8 fragment of this laminin 511. So they are able to produce this recombinant E8 at large scale, and it is still able to retain the full integrin binding activity. So what they found here is that basically the iPSC cells are able to adhere more strongly to that of this E8 fragment when compared to matrigel or that to the laminin 511, the intact laminin 511. So the iMatrix E8 substrate, it can be used for the culture of pluripotent stem cells and has several advantages over the traditional coating methods. So the key features are they are feeder-free and xeno-free. It is consistent. It has this lot-to-lot consistency. So it can be used as a pre-mix or pre-coat method. So in the pre-mix method over here, right, it actually saves the time and also saves the amount of the iMatrix 511 being used. So it is also able to support single cell passaging. So usually in the conventional method, cells will die if the iPSC are dispersed into single cells. So they are usually maintained as cell colonies. However, cells grown on this laminin E8 fragment, they can be cultured as single cells. Also, cells grown on this E8 fragment, they also have a higher adhesion and growth rate, as you can see here when they compare that to the matrigel. So it is able to provide an efficient bulk proliferation, and also it is a lot easier to handle because you do not need to chew the cell culture consumables. Like, for matrigel, you need to chew the matrigel. So this recombinant laminin E8 is widely used in Japan, and it has been cited in many good publications, including publications from Dr. Yanamaka's lab. Now let's move on to look at the 3D cell culture. One of the most common and simple techniques for spheroid formation would be the use of the hanging droplet method. However, the spheroid size that are formed in this method they are often inconsistent and is prone to human error. It is small scale and labor intensive, and although there are several commercially available surfaces that offers properties of low adhesion, the cell differentiation resulting from random cell attachment is still a challenge here. So in order to circumvent these drawbacks, Huckemeyer, a company based in Switzerland, have developed a spherical 5D plate using a patented technology for easy and scalable generation of the spheroids. So the spheroids formed are controlled and uniform size, and it could be used for clinical usage. So basically, this is a 24-well plate with 12 functional wells. So each well consists of a 750 microcavities. So it will provide you with a total of 9,000 spheroids. So the bottom of these microcavities are rounded, which supports regular spheroid formation. There is no sharp edges at the bottom of the plate to avoid any surface-induced non-physiological differentiation. Its patented nano coating prevents the attachment of the cells. It also has sharp edges at the top of the wells to avoid any cells left behind at the top and prevents uncontrolled differentiation. The sizes of the spheroid are controlled by initial staining density, so you can control the size of the aggregates and to avoid any hypoxic damages. So the plate is made of cyclic olefin copolymer, which is suitable for real-time imaging. So this plate is also compatible to the standard automatization processing units. So you can treat your spheroids with drugs and stain for live date stains and analyze the data using any imaging system. So let's take a look at the video here. So this is a time-lapse video showing the formation of the spheroids over a couple of hours. So you can see across the microcavities, the spheroid formation is rather uniform throughout. Okay, so yeah. So these plates can be used in research and diagnostic, in industry, and also even in clinical applications. So in fact, Kokemeyer is getting clinical certification early this year with a human trial for diabetes. So there are many applications that one could use for this plates as shown here. And many cells has also been tested to be able to successfully grow on these plates and form spheroids, which include stem cells. So let's move on to GMP grade recombinant proteins. So when growing organoids from stem cells, the right combination of growth factors is really important to support the growth and differentiation and maturation of the 3D cell models. So CryoYes, a Taiwanese company, has developed a complete selection of cytokines and growth factors with really high biological activity, high purity, and low endotoxin levels. So this recombinant proteins, they are animal free. They over here as shown here, this is a full list of GMP grade recombinant proteins. So these GMP grade recombinant proteins allow for full transparency and traceability, which is a necessary step for building a cell therapy product. So when comparing to the equivalent competitors' GMP products, the CryoYes has a higher biological activity as shown in this graph here. So here we are comparing GMP TNF alpha and GMP IL-2. So CryoYes, as you can see here, the specific activity is a lot higher when compared to that of the equivalent recombinant proteins from the competitors. Right, okay. So one of the common analysis for stem cells is to monitor the cell health. So Biotium, which is a US company, developed in 2001, they are leading cell life science supplier for high quality and innovative fluorescent tools to help to you to image analyze your cells. So here I would like to introduce you to the Rexazuring assay kit, which is a very simple assay for measuring viability. So the viability, okay, so basically this is measuring, you can measure the viability without cell lysis, and you can measure using absorbance of fluorescence. So this kit is equivalent to Alamar Blue or cell titer blue, but at a lower cost. So this viability and cytotoxicity assay kit uses the calcium AM to stay for live cells and also this ethidium homodimer 3 to stay for dead cells. One thing I want to highlight here is that ethidium homodimer 3 is developed by Biotium as an alternative to the ethidium homodimer 1, which is commonly used by the competitors' assay kit. So this homodimer 3 is a lot brighter than that of the homodimer 1. So also, Biotium also offers like live or dye visible dead cell stains, which basically are able to discriminate between live and dead cells. So the labeling is extremely stable because it's covalently labeled, and it can withstand fixation and permeabilization. And there are like 14 bright and photostable stains for you to choose. So you could do either microscope or even flow cytometry experiments. So there is also Atlantis B for the detection of early apoptotic events. It is also could be used in other combinations to detect the late-stage cell apoptosis and necrosis. So it can be combined with the PI, the use with PI, AAD, or ethidium homodimer 3, or it could be used as Atlantis B alone. So apart from all these assay kits, Biotium also offers a whole range of organelle specific imaging dyes, and many of these dyes can be used for live cells imaging, and some of these dyes does not require the need of washing step. So here are some examples of the dyes, like the dark spot dyes or red dot dyes for staining the nucleus. So we also have calcium AM to stain the cytoplasm. And also the cell bright fix and membrane fix for staining the membranes. So one thing I want to highlight here is that these dyes, they are able to withstand permeabilization and metanophyzation. So it allows both surface staining and intracellular staining. So we also have a range of dyes for cytoskeleton like the phalloidin and the microtubules, and also dyes to stain for mitochondria, so that mitochondrial potential dependent or independent options for you to choose, and also stains to stain for lipids and lysosomes, and also stains for physical trafficking here. Yes, so basically I have come to the end of the presentation and hope that the webinar has been useful to you and offers solutions and ideas for your experiments. So if you do have any queries, please do not hesitate to contact us. Thank you.